we extracted stem cells from our own bodies. We basically stuck a needle into our bellies and sucked out some fat cells, and in the fat cells there's stem cells, and so we isolated the stem cells.
Yeah, and we've been differentiating them into different kinds of cells since then.
Yeah, so what you can do is once you have stem cells is you add certain things that will cause the stem cells to turn into other cell types. So let me share my screen and we'll show you some of the stuff we have right now. All right, so this one is really cool.
That's from yesterday.
This is the neurons. Can you see it?
Yeah.
This is really cool. So normally the cells look kind of like this. They look kind of maybe like fibroblasty or a little bit weird because they're stem cells. But what you can see here, this is something that we're differentiating in neurons. And here's one of the neurons. You can see this long fucking axon that's formed between these cells and other axons that's formed between other cells. Super cool to see something like that because we don't see it in any of the other cell cultures, our control or the bone or the adipose or anything like that. It's really neat to see, these cell forming axons between each other and differentiating into neurons.
Yeah, and it's like they're so distinct from each other. The like stem cell population and then the population of cells that have been turned into stuff. It's a lot of times in biology, you kind of like flip a coin whether it works or not, or you end up having to do a lot of stuff to make it work right. This was surprisingly straightforward and easy.
Yeah, look at these. Look at this cell right here. Super
Oh yeah,
cool.
branching.
Yeah, it branches out like that, forming these axons that offshoot. This one's a
It's a
little less
little gray.
obvious.
There we go. Yeah, now I can see it.
You might be able to convince me that it's not an axon. A lot of these things you can do more complicated tests for and stuff like that. But right now we're just in the early stages. But you start to see a lot more cells like this in the neuronal differentiation media, where the cells form axons or long protrusions that connect different cells, which you don't see in the other wells that we have.
Yeah, it's really cool because I think that one was what one factor we added.
Yeah, that one. So for the neuronal cells, generally people do forskolin and human FGF2. We found a paper where they just did human FGF2, so we added FGF2, and that it worked. And so here is the next one. Now this one's bone. And the crazy thing about the bone is what you see these just like protrusions that start to form, these black clumps that start to form over all the cells. You can see some right there. They just grow off the top of the cells, and it's pretty crazy. Basically, the bone is just like calcium deposits mixed with what? What did you say? It was like collagen or something,
Yeah,
David?
so yeah, some of the proteins and stuff. Yeah.
But you get all the cells forming like all these little tiny bone deposits, some really big, some really little.
And over days, you can like watch them get bigger. It's crazy.
Yeah. It's pretty interesting. And we just got all this little bone. Obviously, this stuff is microscopic. We're trying to figure out, ways. One thing we want to do is just like centrifuge it down and separate out the bone fragments that we have and see if there's ways we can make it adhere to each other, or who knows, something to make it like an actual little piece of bone. That would be super cool. If we could use
Yeah,
like fucking meat glue or something.
yeah. You have like a little something you can hold in your hand.
Yeah. But
Yeah,
we've also tried to differentiate the cells into adipocytes, whatever you want to call them. Um, and that hasn't worked out so successfully yet. I haven't looked at my cells today, but right now, the bone and the neurons are pretty obvious, it seems, and we're going to move to next experiments with those. And after that, we're going to continue to work on the adipocytes and, other interesting stuff. But yeah, it's pretty cool results that we've been really just like chugging along with this research project. And we're editing a protocol. David wrote up a protocol, and we're editing that, and it should be online really soon, hopefully like by tomorrow, the latest. But there's always stuff to do, so we're busy.
yeah. And also, it ought to be really cool to get like like just see how long it goes, like just time. I was surprised how fast they transitioned. Um,
Yeah.
but then it's like, you know, once you go, like, what if, like, what's it look like in a month or 2 months, even, if you can keep a flask going for a long time.
Oh, yeah. I mean, yeah,
But
generally primary cells don't last too long, so I can't imagine they'll last too many passages, or even just like too many for the differentiated ones, like too many, too long. But who knows? Yeah, yeah, that's the one annoying thing about primary cells.
yeah, well, luckily I got plenty where those came from.
Yeah. Um. Hey, David, off topic, but what did you think about Walter Isaacson's characterization of you in his book about Jennifer Doudna?
I don't remember much of it. The only thing I remember is his comment about my accent, which I thought was funny. It's
Where do
probably
I
the
get
most
all these?
poetic description of me speaking that I've ever heard. So that was cool.
Yeah, that's the one thing I like about Walter is he's pretty poetic.
Yeah,
You don't expect it out of him. Not to say,
I've
whether
never
you
met.
should or shouldn't,
Yeah,
but you don't expect it out of him. Well, I mean, before meeting him, you know,
yeah,
like, yeah, he just, like, after you meet him, sure. But I mean, like, without just knowing who he is in the public, he doesn't seem like somebody who'd be really poetic, but he is. Yeah, it's funny.
yeah. Nice.
Yeah.
Yeah,
I
the
get all these cool vintage shirts from thrift stores, Susan. I thrift.
but yeah, and yeah,
Everybody always
I
wants to
like
give us their fat, but
I like growing fat cells gonna market them soon. Yeah, same.
but yeah, no, I mean it's interesting. So the one thing we want to do, we're going to start a stem cell class through the Odin,
Be a lot
that
of fun.
will release in the next week or so. That will be in October. Well, we will teach you how to grow your own stem cells, and if you want, harvest your own stem cells. Um,
Yeah.
but part of the class obviously will send you stem cells, so it's not a requirement to harvest your own. But if you want to use all this fat, everybody's always telling us how they have all this fat that they want to use. Well, your chance will be coming soon, where we give you the opportunity to use all that fat that you want to use.
Yeah, the not the best part of the experiment, but it's really cool. And it's also really interesting to kind of like have a separate part of yourself growing in a different place. It's weird.
Ooh, how much would the class be? The class will probably cost, it's going to be remote, so we'll send all the materials to you. The class will probably cost like around $600 because the cost of the microscope and pipettes and all that stuff. Yeah, it sends you all the materials you need to do the experiments. There's actually something else. I'll be right back.
Okay, so it's just me. Yeah, we don't really do a lot of like making chemicals, so like you know, biosimilar drugs or something. Like there might be some stuff with like gene therapy or something one day, but I don't think we do anything with like how to make your own aspirin. That's sort of
I
chemistry.
actually want to thank, we got a special present in the mail from Japan. Rice crackers. And
Nice.
maybe there was something else included in there. We got rice crackers from Japan. We want to thank Yuta. Won't give his last name. Don't want anybody getting in trouble or something like that. But we got, if you've been following along, we got a special delivery from Japan.
You got the seeds?
No, we got rice crackers.
Oh yes, my favorite.
Yeah, different flavors. I'm pretty excited. I don't know what this one is. It looks, I can't read Japanese. What is this? Oh, has like seaweed rice cracker. Oh, neat.
It sounds very crunchy. I can hear it from here.
But yeah, thank you for the special delivery from Japan to our special friend in Japan. Yep. We have lots of surprises, exciting things coming up in the next few months from the CDC. So I hope you keep following along with everything that's going on.
This is a true renaissance.
Maybe it'll be a mukbang. So
stop it.
No, nobody wants that.
No, please stop it.
But yeah, our other project that we've been working on, if you've been following along, we had some successes. And we hope to talk more about it in the future, but right now it's a secret. We don't want to say too much.
It's going to be cool, though.
Yeah. But yeah, stem cells. Daria, you look sweaty. Were you running?
Yes, I had to walk. This is why I was late. Because when the air sirens are on, if you're in the public transport, they're just like, walk, like, we can't move, go walk. So I had to walk, like, really fast, like speed walking. This is why I'm so sweaty. I was just like...
You didn't run?
Like, it doesn't make sense. Like, it's not going to help us.
You didn't run for our live stream?
What?
You didn't run for our live stream?
I did. This is why I'm sweaty.
Quickly.
I was doing it's like 96 degrees in Ukraine, so I was doing my best.
Right on.
I was running up the hill, so
literally running from the government to all your friends. We miss you.
Yeah. I look forward to you getting out of there so you don't have to deal with air sirens anymore.
Yeah, you get used to it. You're just like, oh, okay.
Till one day it's too late.
Yeah, but like,
they're studying for the course. For what? For what course? So for Biohacker Planet conference we have coming up. I think there's like 6 tickets left. Woo! If you can't afford a ticket to Biohacker Planet, contact us, and we might be able to help you get it for less expensive. For the stem cell class, we haven't released it yet, but we plan to release it in the next week. And it'll be all remote, so you don't have to be here to take the stem cell class. You were drunk and you saw last night. So just pay attention on social media or wherever, and or sign up for the Odin mailing list, and
That's exactly what I was about to say.
you should hear about our when we release the stem cell class, but it should be really soon. So be really cool. You're gonna learn how to grow stem cells, how to differentiate stem cells into different tissue types, and do all sorts of cool stuff that's never been done before. I mean, the number of PhDs who's even done this is like, damn, right? Grown and differentiated stem cells. You're talking like literally thousands, like 1,000 people in the world have done this shit. And like you could be one of them. How crazy is that? Right?
Yeah.
In your kitchen, you could be growing and differentiating stem cells. That's how
And
easy
you
we
can eat them.
Now with
Why does
the
it always come
evening
back to
again.
that? Does it always come back to that?
It's fun.
Would the bone cells be crunchy?
Ooh, extra
It's always
flavor.
hot in Austin, by the way. It's
Yeah,
always
it's
hot.
several days this week though. It's dipped below the high's been below 100. So
Yeah, fine.
yeah, I know it's cooling down. It's like 40 days where it's like 105 is like the average.
Jesus, it's 95, 96 here, and I'm dying. Like I am sweaty. I was red. I
Yeah.
was
Luckily, everything here has air conditioning.
Luckily, you're under that fluorescent light, Daria, that really just makes you all pale. So
yeah. That was on purpose. I'm just really white. Like I need to tan because like I'm honestly this white.
stem cells. If you eat stem cells that are like the problem is that I can't tell if you're joking, Jameson. It
Yeah.
won't get to your brain, or right the
Yeah,
prions in
and I
your
mean,
brain.
you'd have to like they'd have to be exposed to prions, or prions that have formed spontaneously, which would be really unlikely.
Yeah, if it's your own neurons, you
Yeah, exactly.
probably
Like
nothing's gonna
exactly if
change.
your stem cells are already infected with prions, you already have it, so it shouldn't be an issue.
Yeah, you
Dude,
probably.
I had a phobia when I first learned about prions. I was in my first year of university, and our professor was talking about it, and I couldn't eat meat for a week.
They're pretty
I was like,
scary. Prions are
I'm gonna
scary.
I'm gonna
Yeah,
die.
yeah,
Yeah,
the whole like Creutzfeldt-Jakob's disease. You're just like, damn. But then you realize it's only if you actually eat the brain of the animal, and then you're just like,
yeah,
Yeah,
like
I usually
I don't
don't
go
do
around
that.
eating sheep's brains and cow's brains, so I think I'm pretty okay.
yeah, yeah. Most of the meat I eat is like leg, shoulder, stomach, belly kind of thing.
Yeah, you can tell this will need extensive. Yeah, probably. Stem cell class will probably need extensive disclaimers like do not try to inject these stem cells into your cell. Do not
Yeah,
eat them. Do not do all this stuff with them. Yeah.
Boring.
I mean, the thing is that like stem cells aren't magical. They're not gonna like cure all diseases. They're more of like a biomedical research tool, not like we're not trying to like cure diseases using them,
yeah,
So
yeah. And this is all definitely like just supposed to be educational and also just fun.
yeah.
Like I definitely never expected to be growing and differentiating stem cells, and like because I mean I didn't go to school for any of this
Yeah.
stuff, so
Yeah. Me neither.
yes, you did.
I mean I went to school for other stuff, but
I mean biophysics. I guess it's not exactly the same, but still way closer than my GED.
Oh yeah. Maybe.
But yeah, but it's really cool because it's like honestly surprisingly doable, Which I mean, I guess it helps that we already did a bunch of stuff for the like human cell engineering class because that already like solved a bunch of the stuff that we would now be having to solve if it hadn't been.
Yeah, I know. It's interesting that I was talking to somebody earlier today, and it was like you know, when you're learning science and biology and stuff like that, it's like difficult when you start, right? All can, like, people, they start a research project and what they want to do is they want to like make a glowing plant. So the first thing they start with is like trying to make a glowing plant, when what they should start with is like trying to grow plant cells and then trying to engineer plant cells and working their way up to this place. And I think that's really really important, that like the progression of the stuff we do, you know, how it's like growing just any sort of human cells, HEK cells in a kitchen, right, in very minimal conditions, then growing primary tissue culture cells in very minimal conditions and understanding how to work with them and stuff like that, and then growing stem cells in these minimal conditions. You know, it's a progression. And if you just start with stem cells, I did act, you know, I grew some stem cells a long time ago, and it was really difficult. It was just a different experience. I wasn't used to growing stem cells. And so, um, yeah, but it's also just, it blows my mind that L15 and F12 work with every cell type.
Yeah.
They don't need CO2.
Yeah. And it's interesting because, like, you know, like how much, like I remember I tried to do primary tissue culture, I mean, a long time ago, like 2017 or something, and just failed miserably. Everything got contaminated. But after, you know,
Yeah.
some practice with the HEK cells for a while, no problem doing primary cell culture. So yeah, I definitely agree. It's a, you know, you gotta grow your skills. You gotta invest in your ability to like do things starting with the fundamentals.
Yeah, I have a
Yeah.
different problem.
Yeah.
Like
I just,
when
you
I
know,
first had HEK cells, I was perfect. Like everything was perfect with my cells. And now like when I try to do anything like streak bacteria on like petri plate, it's like heavily contaminated. It's like it's horrible. And like I'll puncture agar and it's just like it's not good.
So that's a real thing though. That shit happens, right? It's like it's like you get to where you're like, I can, and you just get like you put less effort into the little things, and you're like, meh. And I've definitely been like, I'll slap this subcloning together. I've done lots of subcloning. This won't be a problem. And then just like you half-ass things and you shorten things and skip things, and then it doesn't work. And you're like, okay, no, I have to really do it right.
yeah, for sure. But yeah, I look forward to the day where people stop using CO2 incubators to grow cells. I just don't get it. And like, I don't know who I talk to. Maybe I should write a letter to like Nature or something,
Yeah.
or Science Magazine, just saying like, why does everybody still use CO2 incubators? It makes no sense.
Yeah. Yeah. And yeah, it's weird. It's really weird. And I mean, there may be some cell types we haven't tried that just don't like it or something, but so far everything's been fine.
Everything. Yeah.
And L15 is not even new, right? Like this stuff is been
No.
around for decades.
Yeah.
And it's not
Yeah.
magic. It's like, oh, we need pH buffering. Okay, we can do that chemically. We don't have to do it with dissolved gases.
People have been doing pH buffering with buffers for forever. Like,
No,
this is not
everything
the first
else.
science
Like,
experiment.
I know, like we pH buffer all kinds of stuff for other organisms, and we don't grow any of them in CO2.
Yeah, it's really strange. Like,
Really
who
weird.
came up with that to complicate things? I mean, sure, if you want to say like, is CO2 better than non-CO2? You might be able to argue, sure, because like the CO2 dissolving is like a constant homeostasis kind of,
Yeah.
right? So like you're getting some sort of balance between like the pH and the CO2 dissolving at like all times. But the question is like, do you need that?
Yeah, exactly. Like, do the cells actually care if it's that fine a degree of control?
And like we're using some pre-made media L15 that like probably isn't even really made for this issue. Not as good as it can, F12 maybe a little bit more, but it's like you could probably make it so your own buffered system is just strongly buffered so that the pH doesn't change like ever.
Yeah, and I mean, obviously, most of these cells don't seem to care at all. So, yeah, it's really weird. It's really weird that the dogma that gets absorbed in scientific methodology.
But like, who do we
And you got to
tell?
wonder, like,
On like science Twitter can we get to like?
Is there a manager I can talk to?
Can I speak to the manager?
I'd like to speak to your
The
manager.
thing is like biohackers
Can I
Yeah,
speak to the manager of
in like
science?
5 years, 10 years, biohackers are going to be doing all this cell culture, and everybody in academia is going to look at them and be like, "Wait, what? You don't use CO2 incubators? How?" Like I didn't even know it was possible
100%,
until I started
100%.
trying to do it on my own. And I kept asking, like,
Yeah, see,
why
that's
is there
100%
CO2?
what I think.
And you ask somebody in academia, like, why do they use CO2? And some people will be like, the cells need CO2. There's like this common dogma misunderstanding that like the cells need CO2. They don't. Like that's what I thought in the beginning also. And then I was like, wait, it's
Yeah.
to like buffer pH or something like that. But then I was like, wait, couldn't you just buffer pH other ways? And so I looked for all these medias, found L15 and F12, and like the rest is history.
Yeah.
And
Not
it's
to
not even
mention-
that like invented this shit. It's just trying to popularize it.
Yeah, and that's what I think will happen. I think, like, you know, if people all over are doing this for long enough, you know, like eventually some of these people will be like, you know, teenagers who eventually go to college and become academics, and then they're like, wait, why are we growing this in CO2? I've been doing it in my kitchen since I was a teenager.
Yeah. Right?
I can say that.
I don't read that one. I know you do.
All right, yeah, what? I guess you.
I was
you're still
17
young.
when I started doing biohacking.
Nice.
17.
Yeah, I was much older. Can you read that top one, Daria?
What's that?
Can you read that top
No.
comment?
No, the person just says random shit. Don't read it.
Oh, okay.
Yeah.
What says wrong? This?
No, he's talking about the
I don't know.
comment.
There was a
Oh.
comment in Cyrillic.
Oh,
or
it's just random. Just someone who uses Google Translate and thinks things make sense if they just put it straight into Google Translate. At first, he asked me about my marital status, and now he said, "I don't like David." LOL with a question mark at the end.
I don't believe that. It's too many letters.
Hey guys, hope you are okay. Do you use the same method that break down other cell walls? Would be the diatoms. That doesn't. No, it doesn't make sense, Joe Robinson. Sorry, I don't know what you're referring to or what you're talking about.
Yeah, I mean,
Yeah.
we're like with this, we're working with animal cells, so there's no cell wall, just cell membranes.
Did you miss the reveal on the fat to stem cell culture results? Yeah, we talked about it a bit in the beginning. It's been good. We've been able to differentiate the cells. So, you know, we're 99% sure that these cells are stem cells. Well, they're mixed populations. Again, you know, we've talked about this before. And you have to understand, whenever you're doing primary cell culture and taking cells directly from the body, you can't be like, stick a needle in and be like, only take out stem cells.
Right.
Right? And so you stick a needle in and you take out stuff, and then you culture that stuff, and in there's going to be different populations of cells. And you can isolate and try to enrich for certain cell types, and that's what we did. Enriched for stem cells. And
stem cells have an application because they'll spontaneously differentiate. So even if you did start with 100% stem cells, it'll become a diverse population over time.
Yeah, for sure. Right? Even little small stuffs like pH can, and temperature and things like that. It's not uncommon. But yeah, so we were able to differentiate them. And so, we're pretty certain that these are stem cells. And so the next steps are to make a bone sword and make a brain.
Right? You make a brain, I'll make the bone sword.
I'm really interested though with this bone stuff now because
Right?
I bet you the density of it's different than the cells. So if you spin it down, you'll be able to take off the cell, separate the cells from the bone.
Oh,
And
sure.
then I imagine there's got to be some way you can cross link or just harden, connect it all together so you can form a little piece of bone. There's got to be, right?
Well, there is definitely a lot of protein in it, right? So we should be able to cross-link it with, like you were talking
Cross link
about, with
little
like meat
bone
glue.
pieces to little bone pieces. Yeah, I mean,
Yeah.
there's a lot of cross linkers out there that aren't just meat glue, that are actual just cross link amines.
Acid stuff, yeah. Oh, and also,
So
we might be able to just put it back in, culture more cells on top of it, because they use those little starter sort of almost crystals as little nucleation points to grow from. So if we have a bunch of them clumped together, imagine they'll just stick. But we'll have to test it.
Interesting. Yeah.
Yeah, and I'm also interested, if we figure out a rate, is it exponential, as the cell population increases, or is it fixed by the amount of available, nutrients or calcium in the media or whatever? Is there ways to tweak it to make it grow bones better and faster?
I mean, there's got to be a paper on that or something, like calcium concentration versus bone formation. You'd think.
For sure. I'd be surprised.
I don't know. Maybe not. Maybe that's something we can figure out. But yeah.
But yeah, I'd love to grow
don't ask questions to ask questions.
like a whole piece of bone.
Yeah, right. What I imagine is you centrifuge it down into a tube, and you got this little bone thing, and then you cross link it, and it turns into a little something
Nugget.
I can't see my fingers. Yeah, this big, and it's just a hard little disc of bone.
Mm-hmm.
But I wonder, you have to grow a lot of cells. You'd probably need a bioreactor. What?
Yeah.
Yeah, that's one of the next steps we're going to do is the cells in solution. Yeah.
Even just in a culture flask or something, Even when if it's not quite on the scale of a bioreactor, but if you do it in a culture flask and maybe just keep separating the bone out and putting it in and starting new culture, maybe it takes a long time and a lot of cells or something, but if you could grow a big chunk, if you had a chunk this big, I imagine a lot of the bone will grow on it rather than just random points, because if there's nothing to start with, they just grow a bunch of random starter points. It'd be really interesting if they'll if you can just continuously expand the bone culture after culture.
So they probably do it, right? It's probably like making salt, where they secrete calcium and other things until it precipitates out a solution. So it maybe bone chunks next to it, because there's already a high concentration of calcium, then the salt crystals, it'll crystallize on it, but who knows?
Yeah.
Interesting.
And obviously when they're growing a person, they have some way to grow it in a very controlled way where it grows. Or when you fracture bone, they'll fill in gaps or whatever. So, and they don't just, at least when they're behaving normally, don't just grow random chunks of bone in you. So
yeah,
it would be really cool to be able to grow something you could mount on a plaque on your wall or something.
Yeah. Yeah, if you want to ask a question about something, ask a question about gene editing. Don't ask a question to ask the question. Just do it. How should you start your own molecular biology experiments as a hobby from Japan? There's actually a biohacking space in Japan. I think it's in Tokyo.
And you can also watch my podcast about how do you become a biohacker on YanniWeb YouTube channel.
Nice.
Yeah, I think there's a biohacking space. Jikong Chan. I think he is working on it. Look for him on Facebook or in the Biohacking and Genetic Design Facebook group. He posts in there a bunch, and talk to him. I'm sure he'd be happy to help people in Japan get into biohacking. Um, is it difficult to make a deletion in E. coli CRISPR? A deletion, do you mean like a knockout, or do you want to delete a whole gene? A knockout is totally different, and it's generally, yeah, you could use the lambda red system or other systems. CRISPR probably will be the easiest. The problem will just be screening for it, but that's not too difficult because you can just, you know, do CRISPR experiment, select for the plasmids, and then just sequence the gene. Sequence the gene of like 10 colonies and check for one that just has a mutation. And the majority of mutations that happen in genes are negative. So
you have good odds of getting a knockout, even if it's not exactly what you planned.
And there, you could read stuff. There's a lot of stuff online about doing knockouts with CRISPR, targeting, obviously bacteria don't have introns, but targeting the beginning of the gene. Generally, people target the first intron. But because bacteria don't have introns, you just target the beginning of the gene, because obviously if you just make a mutation in the beginning of gene and it adds one base pair, the whole rest of the gene's messed up. And so...
Yeah, you can do frame shifts, you can add a stop or whatever, but you can also combine lambda red and CRISPR. Like there are systems out there, like the Odin's CRISPR kit uses.
CRISPR combination is probably more difficult than it needs to be.
Yeah.
Maybe.
Yeah. All you have to do is just keep cutting that spot until something changes, which stops.
All you have to do is just transform the CRISPR and the gRNA and Cas9 gRNA and just it'll cut it enough that it'll mess it up.
Yeah, you can't keep cutting the same site forever without something going wrong eventually.
Yeah, for sure. Yeah, that's why it's bad to have Cas9 constitutively expressed generally, especially when you're targeting a gene that is required for survival, because it'll just keep cutting it and eventually it's gonna knock it out.
Yeah, I definitely think knockouts with like knockout is what CRISPR is best at. Like people use it for a lot of other stuff, but it's all way harder than knockouts for CRISPR.
Yeah, for sure. Yeah, because it's just it's just cutting it, and then when it puts it back together, sometimes it messes up. So you don't really have to do anything besides make the gRNA, and that's generally the easiest part and really inexpensive, because depending on what you do, but you can generally do some sort of mutation or rolling circle amplification to change the 20 base pairs that constitute the binding, is it the tracer part or is it the CR? I don't remember, part of the gRNA that bind the DNA. It's 20 base pairs. So
yeah, and most companies out there will make you a custom gRNA plasmid for nothing. It'll be their standard plasmid. It makes a gRNA, and then it'll just they'll just tell us what 20 base pairs you want right there. We'll do it, and it's I don't know, cheap.
Daria, you look tired.
Why do you think so?
What time is it?
You're just sitting there
Oh, sorry. It's 8 p.m. But I was at the gym, so I'm beat.
Oh yeah.
Growing some muscle cell culture in vivo.
In vivo. That's the most complicated way to describe working out I've ever heard.
We'll see who can recruit more people to grow muscles. You doing gym work or David with his follistatin?
David.
Hey.
That's what we should just say. whenever people are like, David, how's your follistatin plasmid going? Just be like, do some push-ups.
Oh man, I'd get way bigger if I had to do
Ooh, look at those guns. You better put them away. Russian soldiers are running when they see those
Careful.
guns.
Yeah, but yeah, it'd be interesting, to see a couple of different ways. How's your stem cell culture going, Daria? Have you looked at your explants?
It's not looking good. I mean, I have them, but because there's still a lot of fat in it, they're just floating on top. So I'm gonna redo it again. I'll try to do an explant just because it's interesting to do it. But also,
Yeah,
I
I think
got some
explant is interesting.
enzymes.
I want to see
I
that.
got pronase and trypsin. So I'll try to do like
Nice.
I do it both ways. I'll do an explant, and I'll just do my I'll do the digestion of the cells.
Yeah, I want to try to do an explant.
If floating's an issue, we may want to lower the liquid level enough. You have to be really watching.
But
make
they
sure-
just dried out when I did that. 'Cause
No,
I
with
did
the explant, I think, yeah. With the explant, you just should remove the fat. What you should do is just shake it up, right, to dislodge cells.
Yeah.
Shake it up a bit, let it sit a bit, shake it like the same. If you were doing it with collagenase or something, but I think like generally you can't use the fat for the explant. You don't want to use the fat for the explant, right? Because if stuff is like the cells aren't going to be like on the surface of the fats, right? They're probably going to be all throughout it. So it's just like try to disrupt it as much as possible, pipette it up and down, do all the stuff to like break it up, and then just centrifuge it and pull off the fat and then put it in the flask. I think that's probably the best because it's just what you're trying to get, and there should be cells. Like what you're just trying to get is like when we do the chicken cell culture, right? Like collagenase is great, but if you just like shake it up, it dislodges a lot of cells. Yeah.
I'll try maybe to add a little bit of like pronase or trypsin to see how it goes.
Yeah.
But yeah, they're like I can show you how they're just floating. Like it's kind of gross because it's like literally my fat because it's like it's in the shape of the inside of the needle, right? So it's like little sausages.
Oh.
Oh. No thanks.
Oh. Okay. That's weird.
Y'all are pussies.
Oh.
I shot myself with a needle, so don't be any of this. Like, you're so
tiny,
awesome and
Daria sausages.
All right, we're waiting to see. Yeah, little Daria fat sausages, which is gross. Look away if you're easily grossed out. Oh man.
Yeah. Daria fat sausages. Looks like you might need to change media.
Focus. Let me just. Okay, are you gonna focus or not?
I want to focus on your face. Maybe if you move out of frame, it'll.
Yeah, just hold it in place. If you just hold it in place.
There we go. Yeah.
Oh, yeah, you can see stuff. Oh, gross. I don't want to see that.
Ew, you see that?
Yeah. Yeah, don't shake it. Don't shake it. Just let it sit. Just let it sit. Don't shake it.
Yeah, I could see, like, I could definitely see him, like, looping through.
Yeah. Okay, no.
Oh, wow, you're looking away.
I don't want to see that. Oh, God.
I'd say it's interesting, though. Like, obviously, if you have a lot of fat cells left, you're going to end up with buoyancy. But I wonder.
I mean, you might be able to just. What I would do is maybe. Do you have any 12-ball plates, Dario?
Yeah.
Yes, a shit ton of it.
Yeah, then you need to use 12-ball. Don't use a flask because it's like it's too big. You'll have a difficult time. But what I would do is maybe just take that piece of fat and like remove it trying to disrupt it too much, put it in like a 12 well plate and just macerate it and pipette it up and down and just like, because the cells that are in there are probably still alive. They just have nothing to adhere to,
Yeah.
right?
Yeah.
And so if you can get it and like break it up more, you might actually be able to get cells to adhere and grow if you just.
That's kind of
Okay,
what
I'll
I
do
was
that.
thinking too. You
Because
know,
you are
There
so
might still be
grossed
cells.
out. Like, what's the
Yeah.
difference when you have adhered cells? They're still like from your belly versus when they're just like floating.
Don't ask me. Like, I don't know.
Such
I put the
pussies.
needle inside myself without any anesthetic. You know, like I
use
can't
anesthetic.
explain. I
You
can't
didn't use
explain
anesthetic?
why. No,
No, just
I can't
Crazy.
explain why that's not gross, but seeing little fat stuff is
oh no,
gross.
it was definitely gross. Like when you were doing yours, and it's like you got the syringe pulled back, and it's like, like a little.
I know, and it's like I was doing it, and then like the plunger went back into like because of the vacuum, and
Yeah.
you're just like, oh. Like I was fainting in the end. I couldn't feel anything because I had Anna. She has a nurse background in our lab, so she had lidocaine, so she did like a professional work of doing it. So I didn't feel anything like at all, like completely didn't feel anything. So it was
Yeah,
weird because I was like sticking it in a butter, like the needle. But then at the end, I was like, oh, I feel lightheaded.
Yeah,
yeah.
yeah.
You don't
I
have to
didn't
tell me
feel
what's
any.
gross or not. I know it's
Yeah.
gross. Like, come on. Like,
Christy, you're a savage.
like you're not gonna. I don't know what it is. It's just like some stuff. Like when if somebody like vomits on TV, it makes me vomit.
Yeah.
It makes me gag. I don't know why. I'm like one of those people who just like some stuff. I don't know. Like I'm usually have a pretty high tolerance, but some stuff
Yeah,
is just like.
that's surprising because you're pretty gross.
Yeah, I think everybody's got stuff that's sort of ticks there. That's gross box.
You know.
Yeah.
But yeah, I think it's a good idea though. Like take those, like, you know, pipette the crap out of them, put them in a 12 well plate, try and concentrate those cells up.
Yeah. I'll
Yeah,
do
but I
that
really
because
want
they're
to.
not dying. Like they're definitely not like.
I mean, the media's changed color, so something's happening in there.
Yeah, well, I changed it, but like yeah, it's been like 3 days since I changed it last
Yeah.
time, so it's like yellowish.
Yeah, we're doing ours on like Tuesdays and Fridays.
Which I forgot to change my
Media
media yesterday.
I
left
even
it-
left it out, warmed up, and everything.
Yeah, I stuck it back in the incubator, and it's probably all not good because 37
Oh
CO.
no, it's still in there. Here.
Probably, yeah, I totally forgot.
It's totally in there. Well, if it's full of fungus and bacteria, we'll know.
Well, show us.
Or
I showed
something.
my fat on the stream. Now it's your turn.
Oh, you want to see some of ours?
None of our fat is left.
Oh yeah, true.
The fat's gone, but the I mean, I can show you the plate. Not that you can see anything unless I wheel around to this other computer and show you them on the microscope. Yes, I'm still breeding dogs.
Oh, David, I forgot to ask, how's your follistatin project going?
I'm not doing push-ups.
David gets the most questions, and it's always those same two questions.
Well, I mean, and I'll just kind of answer it. You know, like this stream, you know, we have a topic, so we'll stick to that. If you got questions for me about that, just look on either my YouTube, Odyssey, or my Patreon.
Go to David's Patreon, subscribe to his Patreon. You know,
Yeah, David
throw a little
posts
money his
content,
way.
not like
I'm
me.
sure he'll
I don't.
answer all your questions.
Yeah. Otherwise, you know, we'll end up spending half an hour talking about something that's only relevant to me.
Yeah, if you, I think it's a good idea. If your questions are not relevant to the topic of the stream, ask them on Patreon. Pay a few bucks, but you'll get an actual answer.
There you go.
So yeah, if you want to know about my marital status, please subscribe to my Patreon.
Hey, you know,
That's a good one. I like that. You know,
Yeah, me too.
Yeah.
That's
if you want to like try to date,
you're
try to think
gonna be
on
weird.
first.
Please pay to be
Find
weird.
out.
Yeah. And like I'm not putting it out on like YouTube and everything, but you can
Yeah, don't be
probably
weird and cheap. You know.
Exactly. Don't be weird and cheap. It's never gonna work. You'll only be weird or
worth
cheap,
it.
Yeah.
not both. Yeah. Hey.
Here's mine. The thing about 12-volt plates is they're the squeakiest things.
They're horrible. I agree. I also hate the condensation on them.
Yeah. So annoying.
And like I
Condensation.
used to, like it's good this time. I have some cells in a 12-well plate, but just I used to always get contamination in a 12-well plate.
It's kind of a toss-up for me for
Saw the documentary on Netflix and it was great. Glad you enjoyed it. I was just myself, so I didn't do much. Actually, there's a lot of work doing documentaries. By the way, Biohacker Planet documentary coming along. I've been editing a bunch of footage. If you want to watch it, come to Biohacker Planet. The conference that we're having at the beginning of September. It's going to be cool. A lot of fun footage for sure.
All the cool kids are gonna be there. Us.
Speaking of which,
We're the cool kids.
I wanted to shoot your portrait again, Daria. $200.
I mean, honestly, I don't think OnlyFans is a bad idea. No, don't get me wrong. Like, but yes, no, I think it's a good idea. Like, come on. Like, just show your boobs and get like so much money. Like,
wouldn't work good for me.
oh, David.
Yeah, what's that noise? Oh, stop. We
Okay,
can't see
who would
it.
subscribe to my OnlyFans? Hit a plus in the comments.
No, not really work. Put a plus in the comments.
Just gonna point one computer at another one. Okay.
It's like a Rorschach test, you know, when you look at
I
the
know.
picture and you try to guess what it is.
Yeah. So this one is this is a lot of my bone cells. You can see like this big clump right here. I've been watching it grow for a while. But yeah, so it's like a bunch of little ones. Oh man, it's so hard to do this backwards because I'm watching it on this screen. But yeah, and then you can compare those to like some of the fat cells I've been trying to induce. And you see there's like no clumps of bone in them. I haven't done neurons with mine, so I just have like negative control.
Some nice looking bones. Some nice looking bone structure you have, David.
Well, you know. Okay, so this one.
Crunchy bones. David, can I eat your bone cells?
Well, we'll have to discuss payments.
Stop it with the get
Just
weird.
trying to like.
Are you
think
starving
Daria's
over
hungry.
there? You living on top ramen?
Just trying to you know, just putting some ideas for my OnlyFans.
Biohack the Planet will not be streamed because it's workshops, and so like it doesn't make any sense to stream it. So unfortunately, it probably will not be streamed because like, yeah, it'll just be really difficult to stream workshops, hands-on workshops. You need like active camera person.
Yeah. But yeah, like
If you
you can
want me
see.
to make a vlog of Biohack the Planet,
What are you trying
hit
to
a
show us,
plus.
David? Are you done yet?
Yeah, I'm done. But
Or are you
this
just
is,
gonna leave the computer screen up instead of your
Yeah.
face for the rest of the stream?
But yeah, so that's what the like negative control looks like. So you can see it's like super different. Okay, let's see if I can make more loud squeaks for Daria. I know you love the loud squeaks.
How's it going, the physical improvement? Well, I got boobs. I don't know if that's... Ah, dude! Oh, God! Come on.
Can we ban David from streams now?
I don't know. That was pretty rough.
Yeah, it's
I don't even know how
bad.
you made that noise.
Huh?
I
I
don't
just
even know how
rotated
you
the
made it. It's just. Ooh, I think you damaged everybody's eardrums in that one.
laptop. Apologies to anyone wearing earbuds.
Anyway. So you can't be on OnlyFans till David grows boobs.
I'm good.
What?
That would be funny.
If David get boobs?
It would be really unexpected.
And everybody in CDC would have boobs.
Well, all I have to do is just keep eating enough pizza, and I'll eventually get some.
Anyway.
But yeah, I do look forward to Biohacker Planet. I'm really stoked about it this year because I think the
for some reason your volume, you can't hear you as much,
Yeah.
David. I don't know if
what if I just get closer?
you're so far away. A little bit, but still it seems a little muted. I don't know what happened.
Let me check my settings.
We have to try to turn up your mic a little bit.
What about that? Does that make any difference?
Yeah, for
Yeah.
sure.
No.
Okay, I switched it to the mic, to the webcam's microphone.
All right.
But yeah, I'm excited about Biohacker Planet this year. I like the idea of workshops. I like the idea we're gonna, get to actually like teach people stuff instead of just stand up there and talk about stuff.
Yeah, so do I. I mean, talking has its place, but like, I don't know. I think that like biohacking, the where it is right now, a lot more people would enjoy actually doing learning hands-on stuff than, just like listening to people talk.
Yeah, of course.
Because it seems like there's a lot less people who are doing the stuff that we're doing, and a lot more people who are willing to talk, and we want to change that.
Yeah. Yeah, and I think it's like there's a lot of things that are really difficult to learn, like in a not hands-on way. Like for me, I know there's a lot of things like OD 600, right? Like you're just transforming bacteria or something. You want to see like, oh, is this liquid culture, dense enough? And it's like if you don't have a spec, like you don't need a spec, you can just kind of eyeball it and it's close enough usually. But like if you have no experience and there's nobody to tell you that's dense enough, it's really hard to get those little subtle things. And then it's like, you can do little crazy things like through like pipette gently. Well, what is that really? How gentle is gentle? Like it's way easier to like watch somebody and have somebody like, no, do it a little more like this. And it's such a faster way to learn.
Yeah, for sure. Yeah.
Otherwise, you just have to double trial and error like I did.
Conference. There's no Tony Robbins coming or other people like that. That'd be kind of funny if there was, but Tony Robbins, genetic biohacker.
Isn't he? Am I thinking about a different guy? Is he the like personal motivation guy?
A bulletproof coffee?
Yeah. No, that's the different guy. But this guy like is like.
Yeah, he's hype.
Like,
he's a hype guy.
Yeah.
First he's like, how many people out here suck at life? And everybody's like, and he's like, yeah, you do suck at life. But then he's like, but you know what? I'm going to tell you the secret. You don't have to suck any longer. Just follow these 5 simple rules, and your life will be changed.
Yeah. Yeah, it's funny. There's like a whole industry around telling people like, you don't have to suck so bad.
Yeah.
Just try.
I mean, people pay for it. So
yeah. That just tells you like how rare like encouragement must be in the world. Like if you had to pay a professional to tell you it's okay, I believe in you. Like just $2,000 for that life lesson. Yeah. I would hope people would have, friends and family. Be like, you know
We'll
what?
start doing that. We'll combine it at Biohack the Planet. I will make fun of you. I'll tear you down, but then I'll build you back up. I promise.
Okay. David,
Hmm.
I forgot to ask, how's your fully statin project going?
Oh,
No,
God.
and also, when are we gonna get the glowing puppies?
Oh, I know. Tomorrow at noon.
I mean, at least
I
you
know
get
it's weird
decent
how
questions. I get
like people always come to ask David these same questions. Like nobody comes to like ask me or Daria any questions, but people like specifically come to ask David these like 2 questions. Like that's
Well,
it. Really funny. I don't know what
some of
it
these people are
is.
probably coming to my YouTube or my other stuff, but also like I guess because I have these like really long-term projects and people are impatient for me to go ahead and finish. I mean,
This
I
is
am too.
funny. It's like what about me and Daria? Like we feel left out.
I'm jealous because like I get questions like, "Are you married?" These your questions are about science.
I get questions of like why are your nipples so big?
I saw that one right there. Oh,
So, we're all doing our best.
yeah.
Somebody the other day in person told me that my CDC tattoo was super cool.
Nice.
Cool, I
Yeah.
have one too.
Yeah. Now
They
I feel
said
left out.
good for health, bad for education. They're like, that's awesome. I was like, yeah.
Yeah. I still
It's
get
I
such a
never been
nice
complimented
tattoo.
on it before.
I'm just gonna
I
get
always
mine
get
right on the forehead.
people talk about all my other tattoos. Everybody talks about these soot sprays because it's like the basic bitch tattoo. Everybody's like, oh, I know the movie that's from. It's from, I'm like,
Yeah.
Spirited Away, and they're like, yeah, and I'm like, oh, yeah, it's so great, and I'm like, oh yeah. Seriously, one time I was out to eat with somebody, and two of the waitresses came up and said something to me about.
It's a very recognizable tattoo.
Yeah, basic bitch tattoo,
Well,
man.
it makes you feel better. Nobody's ever complimented me on my tattoo.
I don't think anybody's
Oh, you
seen it. I
never
don't even
show
know if
it?
I've seen it.
No.
I think maybe I've seen it once.
Yeah.
Subscribe to David's Patreon and maybe he will show you
You
his secret
should have just like
tattoo.
one picture in there just for people who are curious of what your tattoo is.
Yeah. Okay, here's a good question. Can we use adult cow serum?
You can, but
Yeah.
it just doesn't work as well as like newborn calf serum.
Yeah.
I know,
You use
crazy
adult
bicep.
human serum.
Oh damn! Look at those guns. Russian army is scared. You got so many guns.
I know.
Yeah, we've even
Careful.
grown cells in adult human serum. So if you really get desperate,
Yeah, I
Yeah.
think adult human serum works a lot better than adult cow serum. So there's
if you're
probably a
going
lot more humans than cows in India. So you got that going for you.
yeah, 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 and it, like if
That was a good one. Beat me up, Scotty.
yeah, but yeah, so you just you can take your own serum if you're growing your own like primary cells or something, and
Even if you're not, even if you're just growing, cells.
Yeah,
Like I
other
use my
cells.
serum
Yeah.
on like all different types of cells. I think even
Just-
chicken cells I've used it on.
Yeah. Just try not to eat like fatty stuff before you draw your blood because your serum is going to be.
Yeah.
One thing I think is interesting, something I wouldn't mind trying is like, what's the like minimum? Like, what's the like how like dystopian can you possibly get and still do cell culture? Like, use your own serum, use like glass or something. Like, it's like you need like a syringe and.
Are you
serum.
trying to do like a steampunk?
Yeah, or like
Gattaca.
what's the minimum? Like,
Oh,
can you fake
okay, I see. Mouth pipette with a needle and a rubber tube.
Kind of, yeah, like that. It's just like,
Okay. You just got like a rubber tube where you're like dripping your blood out
stab
of
yourself
yourself.
and you're just like, look at Darius' face. But no, I mean, like, how minimal can you get it? Because like,
Ooh, I have an idea. I
I
was
can't
thinking
think
about
of how
it.
you
It's really gross, so people who are sensitive to gross stuff do
Oh
not
gosh.
listen. But
it
I
really
was thinking, when you get your period, it's not just blood, it's literally cells because your uterus is shedding from inside. So it's endometrium cells. So why not collect it and try to grow it? And you get it for free like every month. And it's almost painless. Like you don't have to do any like
Aren't most or all the
intrusive
cells dead though?
No, why would they? Because they come in chunks. Fuck yes, period science. Yeah.
I mean, they're what, endothelial cells or something like that?
Yeah.
Yeah.
Because
I
they come
mean,
in chunks and there's still serum and blood and everything. So I think they should be fresh. Fresh out of the oven.
it seems like it'd be easy enough to try.
I need a shirt that says period science. Fuck yes, period science.
I'm sure somebody's tried this before.
Yeah, I mean, I'm sure
If
there's a
someone's
way
gonna make this shirt, please send it to 49001 Dnipro, Ukraine.
if you can get it in.
Like, come on. Like, I think it's an easy way to, and I'm pretty sure no one has done it. So it's kind of like
No,
it's a
I
science.
know.
It's science.
I know at least one artist, Jiao Bao Li, who we collaborated with. I know she was trying to culture stuff from her period blood. I don't know if she ever cultured cells or not. I don't remember, but I'm sure also there's got to be a scientific paper where somebody's tried to culture cells from period blood. I can't imagine there's
Whatever.
not, maybe, but
Yeah. The other
a
issue.
period piece. That's good.
Okay, so when is my period?
You may have to use a lot of antibiotics.
Yeah.
So on
Biome bacteria.
Tuesday, 23rd, who's gonna watch the live stream?
Life stream's on a Wednesday.
Subscribe to her Patreon. The Patreon one.
It's getting weird, y'all.
Put it behind the paywall.
Yeah.
Yeah. I'm not. This content is not for free.
Yeah, but I tell you, it would be interesting because there's some stuff with like artificial wombs and stuff that use those cells as like feeder cells and things like that.
Yeah, we were talking about it like a year ago, but now I actually want to try.
Yeah. I mean, I'm sure there's going to be some unexpected hurdle because otherwise I feel like it would be more common. But
Yeah, I don't know.
maybe it's dumb hurdle.
Really? You feel like it'd be more common? Like,
Well, I don't know. I mean,
what? What? I'm confused.
I don't know. It feels like it'd be something I've heard of before.
Oh, okay.
You Googled.
I mean, I haven't Googled it, so maybe it is more common than I think. But I feel like I would have bumped into it.
Yeah.
Maybe. Yeah. I don't know.
More junk for the OnlyFans.
I don't think it would be that common, but yeah, who knows? I can't imagine that there's somebody, even if it was just in like the 1970s or something, didn't try taking period blood and like.
See, but that's the thing. It's not only blood. This is what most of people, or like 50% of
What would
population,
you like
think
me
that
to call it?
it's just blood. No, sometimes it's blood, but there's
Want me to call it
chunks.
period
too.
liquid? Period.
No,
Well,
there's
it's
chunks.
also like, yeah, clots.
Okay, well, what word would you like me to use to describe the stuff that comes out of your body during your period, Daria?
Well, it's literally like it's
Menstruation?
blood and chunks of your endometrium.
We should start a
It's
campaign
like it's
to
not
change
only
the
blood.
name.
You literally get like
It's no
chunks
longer
like
called period
this
blood.
big.
We want to rename it to period.
And like a lot of women just
chunk
get these chunks of cells that haven't like dislodged. See?
Period situation. Um,
Anyway,
cells have
David,
normal
how is
cells.
your follistatin project?
Yeah, those would be uterine lining cells, so they wouldn't be
Uter
like
juice. Oh,
uterine juice. Okay, that one wins.
it's actually
Uh,
pretty nice. Can I use that name?
I mean, there probably would be a lot of dying cells, but you don't need a lot. You don't need all of them, you know?
Bloody science.
It's a
I've
surprising
already
thing
bled for science many times, so this is
in
not like.
biology.
Yeah.
Like, I remember, I've read some papers with some stuff that is done in the paper. It's like, I would not do that.
Yeah, whatever, man. You wanted to cut yourself open with a scalpel to get your fat out.
Sure. That doesn't bother me. But it's
David,
like,
when we were doing it, like right before we started, David's like, "Are you sure we just don't want to like use a scalpel and like cut open our stomach and like take out the fat?" I was like, "What? Are you crazy?"
that's less freaky to me. Like, I brought my staples and
What? He wanted to cut himself open with a scalpel. I was
because
like,
a hole, you
"What do
just
you mean you
grab it.
wouldn't try this stuff? You would try crazy." Oh, come on, man.
Like a syringe is like in your body. I don't know. I'd rather have a hole than a needle in me.
Yeah. So David, I think he's lying. I think he would try a lot of crazy stuff.
Yeah, but I've seen like, I don't know, I've seen some stuff.
I mean, you just gave yourself biopsy punches to try to grow the cells. Like that's intense, man.
Yeah, so did you.
No, but
That's
I
like way
genetically
more
modified
invasive when you
myself.
use a scalpel.
I genetically modified those cells, and then I had to do the biopsy punches,
Oh, you're right.
run the analysis or whatever. You just like did it for fun, and it didn't even work. That sucks. Oh man,
So,
Oh,
David, you're a masochist.
I know. I think so.
I used it in the
Yeah, I
septic.
think he likes it. Like he likes pain.
I've been accused of
Hurt
that a few
himself.
times, especially back in my martial arts days. But no, I didn't feel any pain because I used lidocaine then too. Lidocaine's wonderful for doing crazy stuff.
Yeah.
It's weird to me that it's so unavailable, Like, is there a big
Lidocaine?
problem with people abusing lidocaine?
I know in Ukraine you have
Actually, we
to
have
have.
benzocaine and stuff. I bet you
I
could
know,
probably
it's
use
been
really
all day
similar.
long.
I imagine it's just because like you can probably kill yourself with it pretty easy.
Well, I'm sure. But like,
Yeah.
But like
so
you can kill yourself with ibuprofen.
yeah,
Yeah, but not that easy.
Yeah, but still.
Like
Yeah, I
lidocaine.
had to like. I walked into like the first
I mean, I'm
pharmacy to
not
see
saying
my.
it's reason, but like yeah.
it's still just a weird thing to me. I did use a patch for the biopsies, but then when I did uh, the stabbing, I used injectable because the patches don't go very deep. Like when I was doing the punches, like the skin would come up, but as soon as the punch got deep enough to like get into the fat layer, which I could tell because there'd be like some fat on the bottom of the punch, or the cylinder, I could feel it. So like the patches are really good for numbing the skin, but they don't really go deep. And that was after leaving the patch on for like 2 hours.
2 hours.
I injected lidocaine.
I
Yeah,
think
the
when I
injection
did
one more
my
time.
biopsy, I just drank some whiskey. It
It's
hurt. I still got a video. I got a video of it, because I took a video, and I'm just the whole time I'm just like, oh god,
yeah, nature's anesthetic.
like this hurts so much in the video.
Yeah, I bet.
No, without lidocaine, I wouldn't be doing it.
Yeah, I probably would if I had to, like if I thought there was some benefit, but like I would not want to. I would much
My
prefer.
body is weird. Like usually after like the first feeling, like if I stab or like poke, usually after the initial like poke, like I don't know, like pain shuts down. It's like a weird thing.
No, I
Yeah,
don't have that.
yeah, I don't have that either. It continues to hurt, especially the more you like
Just
move the needle around.
yeah, I didn't feel pain as much as I just felt nauseous.
Yeah, honestly, like
Yeah.
that's a big thing that worries me too. Is like I have like the first time I tried to use a butterfly, I was like really bad at it, and I was going too deep, and I was like going through the vein and then out the other side a lot, and I'm like hunting around because I'm bad at it. I'm not trained in this
I
stuff,
can't believe you
and,
tried to do that.
and so
I'm
I
trying to
it
draw
took me
my
like
blood.
it took me like 10 minutes, to like fish around. And I was like white as a sheep. I had to take a break, catch my breath.
Yeah.
My adrenaline was just
Same.
like pumping, and there's like nothing to do with it. And eventually you just get like lightheaded and yeah, your
Yeah,
body's like, don't stab, don't do it, don't do it. And you just have to do it anyway. And it takes like yeah, it's no fun. It's no fun.
Yeah. It didn't hurt at all. Like it was just weird that I could like poke and like put the needle so deep into like my fat. But then like I did like I don't know 8 pokes I think total, and then I was like, okay, I need to stop because I was just like I was like, ooh, I feel like I had it, and then I'm like, oh, I'm gonna faint right now.
Yeah, I've been there. But I think, like for me, I just pulled the plunger back and just held it in like the like vacuum position, and then
yeah,
just kind of like
yeah. Moved
fished
it around.
around.
But like the vacuum didn't work that much for me. The best what worked for me is just like stabbing myself and just like collecting cells inside the
Yeah,
needle.
just like
Yeah, but I have a lot more body fat than you, so it might make a difference.
I'm still walking around because I have the dots from the.
Hmm.
Uh, I still walking around with it.
Yeah, I still have a tiny bruise from mine.
I didn't have a bruise, but I couldn't actually feel anything for like 5 hours
Yeah,
with lidocaine.
same. Yeah, and then and it really wasn't bad. Like overall, the pain was nothing. Um, but
No, yeah, I it didn't like if I
yeah,
touched it
totally
with
feel it.
my finger, it just hurt like a little bruise, like not even.
I would do it again, but I probably will do it again because I really want to do some stuff with these explants and stuff, and also just to
Well,
get
I'm doing it again too, so
stuff. Just I want to, and there's got to be like a couple
Some
things. I'd
drawback to
probably
it.
definitely use a smaller gauge needle, which there's something I want to try. You know, um, and like you could probably get away with using smaller gauge needle for sure.
Yeah, yeah. And that was my biggest fear, honestly, was that we
Using
weren't
18
big
instead
enough.
of 16. Yeah, I was afraid it wasn't big enough, but it was fine because usually it just like, so you might be able to go down to 18 from 16 or something like that. I think some people have used 18, so I don't think it would be a problem. Because most of the hurt comes from like stabbing through the skin, at least for me, and then it's just like it's not as bad. Um,
And it's
might be
pretty
able to
big.
get away with a smaller gauge.
It's pretty big.
Yeah, I mean scale doesn't really do well
Yeah.
with.
I mean, but it's beefy, you know. And I was
Yeah,
worried that we'd just stab and then like pull back and nothing would happen.
yeah.
yeah, for sure.
What? Just ignore. Just ignore it.
Is it theoretically possible to cultivate stem cells? You can cultivate stem cells from any organism, every organism, even plants have stem cells. So like you can get stem cells from every organism.
Yeah, of
Yeah.
course, plants have stem cells because they have stems.
Oh gosh.
That was so
Not those
good.
kind of stem cells. Don't listen to her. They have real stem cells.
They have both, technically. Do they have stem cells in their stems? Would that make them stem cells?
Stem cells, yeah.
Oh, let's see.
I
Body
was afraid it would
processing.
not be big enough. Every man's worst fear.
You need to
I
learn,
couldn't find the
some
16
stuff.
gauge needle. I had to buy like a blood infusion, like a whole thing for like the
Yeah,
catheter and
IV.
everything for. yeah, for like IV for when you get your blood, donating blood, and like then take a needle from it and like put it on the syringe.
Mm.
Yes, in the STEM class.
Yeah. Yeah, I
Yeah,
imagine a lot of. I'm surprised you could get a lot of needles and medical stuff. Are people using it? Probably short supply in Ukraine.
yeah,
I don't think
Not
it's a
really.
short supply because like I don't know. We're not really short on anything, but like it's really hard to get like stuff if you're ordering from like Alibaba and stuff like that. Now it takes like 1.5 months to get here
oh
instead
wow,
of 2 weeks. I mean, because there's no planes flying, so it has to be done by sea.
yeah,
Uh, so, but like you can get any lab plastic, but with some regions you had to like, I really like I had to steal these pronase and trypsin. Like I had to text a friend in like some academic lab that could give me the old regions.
yeah. You just have to
Yeah.
grow your own trips in. I wonder. Yeah, I still look forward to you getting out of there though, because you
It's
never know
fun here. It's
where's.
really nice here. Just really hot.
See,
But
just
yeah.
move to the cold, like Austin.
No, it's like it's so weird because like life is normal, right? But there are no planes flying. So if you hear a plane flying, run
Yeah,
because and you hear air sirens, and when you walk around the city, well, you see a lot of military ambulances, and you see a lot of like anti-tank, hedgehogs around the city, and a lot of stores put like plywood on their windows in case there's explosion so it doesn't shatter, and that's pretty much
yeah. Still,
I'm just reading the
Yeah.
comments and I'm just like,
Yeah.
yeah. I mean, yeah, never.
Anyway,
dude, you're gross.
what else? So, what other projects do we have coming up in our backlog?
Um,
What are we gonna do? Oh, IPSCs. That's one thing that we want to start working on also.
yeah,
Turning ourselves into IPSCs, which will be pretty cool.
and that's
Yeah.
induced pluripotent stem cells for anybody who doesn't know.
Yeah, iPSC is induced pluripotent stem cells. So a pluripotent stem cell can turn into basically almost every cell type.
Yeah, and then you
So that'll be
can,
really neat.
yeah, you can grow all kinds of crazy stuff. Like,
Yeah, that's where
all,
they grow all the organoids from, right?
yeah, yeah.
Yeah.
I've seen some organoids where they're doing like brain organoids that grow these little like pre-eyeball things, and it's like a little brain with eyes. It's the freakiest thing.
Yeah. Could you make sperm cells with iPSC? I don't think anybody's made sperm cell. Like, I think you can make. So the way they make sperm is they make the stem cells for the
The cells that make the sperm cells.
yeah. But then to make those, I don't think anybody's been able to do it without the actual cell. So you have to. The only way to induce it is to put the stem cells next to the real cells, so they turn into it. So you know, it's possible, but it's not possible to do de novo yet without any other cells. In humans, I think they might have done it in mice without any other cells, but not in
Yeah.
humans, I'm pretty sure.
That's
I
probably just
really,
figuring
I really
out.
want scientists to do like to make a not fake sperm, but like artificial sperm. That would be
Yeah,
really important.
Yeah, it would be
yeah.
really cool. But there's also I've seen some stuff where they just fuse cells uh, to get different stuff without having like so you can like fertilize without having to use a sperm cell somehow. But I don't know if that's ever been done in a human cell.
What's the easiest method to get a bunch of human egg cells?
big old needle usually.
Yeah, like a really big needle.
I don't know how it's done in humans, but I know I know how it's done in dogs. It's pretty invasive.
Yeah.
I
Could
think
you
same in
create like a
humans.
brick out of bone? Yeah, the thing is, we're trying to figure out. So when the bone is made, what it appears in these cultures, you get some tiny little pieces, a lot of little grit,
Grains of
and some
sand.
bigger pieces. So we're in the process of trying to figure out how to grow bigger pieces slash how to combine small pieces into bigger pieces. Um, I don't know if it's possible to grow in a specific shape. That would be interesting, but that might be a lot more difficult because the bone grows where the cells are. And so, um, the cells will cells migrate, they move. And so it would be hard to keep the cells in one place necessarily and have them secrete in such a way. Not saying it's impossible, but right now I think it's kind of outside of anything reasonable. Like, yeah, you might be able to grow it on some scaffold and get it to kind of do something, but
That's kind of what I was thinking because, like, I know, like, one of the things I want to play with is titanium will osteointegrate. That's why they use it for like bone repairing, plates and stuff. So you might be able to use like titanium foam or something really porous. And maybe they'll like use that as a scaffold, or maybe you can like put down some bone and they'll use that as a scaffold, or maybe um, like you could even control the shape of the adherable surface somehow. I don't know.
yeah.
But as far as like sperm is one of the cheapest and easiest to collect, it's like, yeah, but that's only for about half the people.
Yeah, you
Yeah.
gotta realize that there's people who can't make sperm and they want to.
Yeah.
Yeah.
A lot of people are not caught up.
Are you jealous?
Yeah.
Jealous. Just
Could those pluripotent stem cells be used for chimeras? Uh, I mean, I don't know, honestly. Like, if you just like put some iPSC
Sure, why not? I mean,
fetus, I think it might make a chimera.
yeah. Could you recommend books? I don't think books help with biohacking. I think to do good at biohacking, you really should experiment. And usually, experiments lead you to read research papers if you're doing them correctly, right? So, like for our stem cell experiment, we probably read like 20 research papers before we started doing it, trying to figure out conditions, the way people did stuff and extracted stuff, and all these other things. Um, and so, it should just lead you down to learn stuff. So I would just say do experiments, learn to do experiments. Um, yeah. Dan says, when are you going to use the thing is, Dan, so we thought like getting fibroblasts is kind of like just as hard as getting stem cells, right? So if you want to get cells to like turn into iPSCs, it's generally going to be just as hard whether it's MSCs or fibroblasts or anything. And I actually think because you don't have to do a biopsy, um, getting the MSCs are easier than getting fibroblasts because fibroblasts generally you have to do a biopsy punch, which those hurt.
Yeah.
Where it's like just sticking a needle through your skin, that's all you have to do. You don't have to like pull out a huge chunk of your skin, and that's where all the nerves are. Like there's not much nerves in the fat tissue, so yeah.
And also, but yeah, we would probably start with some of our stem cells and then just transform.
Yeah.
I'm gonna do some testing as soon as I grow more with transforming, just with like GFP, so I can get. We're probably gonna use electroporation or something. Get a sense of like how tricky it is to transform them. I imagine it won't be a problem, but then we can have a sense of how easy they are to transform. Then we can just use Yamanaka factors to turn them into iPSCs, theoretically.
Yeah, exactly. That's what we're working. One of the things David's been working on for us is creating a single shot plasmid that has all the Yamanaka factors on it.
Yeah. So it ought to be pretty cool. And then we can, but like iPSCs are harder to work with than mesenchymal stem cells from what I'm seeing. They're like really subject to
Yeah,
differentiating easily. So we'll have to practice some of that too.
really sensitive. Generally, you do serum free with iPSCs also.
Yeah.
Yeah, iPSCs, what I know is just like really difficult.
Yeah, and they don't like
You know,
trypsinize them to like spread them into new plates. They like slice them up and like grab them and put down little like blobs of cells. You have to be careful that they don't fold or they'll differentiate and crazy shit. So those will probably be a little bit harder to deal with, but you know, it'll be a lot of fun. And we can do crazy stuff with them. I mean, okay, invest in 23andMe now. I didn't.
Did you invest in 23andMe? Mm.
yeah,
Yeah. Let's see what else. What else we got
yeah.
other than that? The IPSCs thing is going to be cool because it can give
Oh
us a
man,
lot of
what's going
options.
on with the internet?
I don't know. I jumped off the main Wi-Fi and I'm doing it over my phone right now. So maybe we're doing the same.
Yeah. Oh, you did? You're streaming from your phone?
Yeah. 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 For
Am I just
whatever reason, the
freezing up
Wi-Fi
all
is
the
really
time?
crap right now. Not all the time. You're
No.
you know, but
Odin,
Yeah, you're freezing.
what are you doing?
but yeah. How's the Warzone science coming?
Good.
Yeah.
Doing some. I have my cells there. The incubator. This is my lab.
Yeah. Well, it'll make a cool story
This is you.
once you survive and get out of there.
Yeah, I will. Like, come on. Relax. It's just war.
Nobody's ever said that before.
Like, you have to get to a certain kind of, like, I don't give a fuck to, like, survive, so.
Yeah, but we care. So
Come here. It's fun.
no, nobody wants a dead Darian.
No. Is my
How will blood
internet
serum pregnant?
not working?
Probably not much. I mean, there might be some different hormones, but it probably won't matter once you get them in culture. I have not used a kilobaser. Oh, are you? Okay, I thought you froze up. You were just really still. But yeah, what else have we got? I think once we get your antibodies can detect HIV antigen, right? I don't think we have anything like that. Oh, it might just be me now. Let's see. I'm going to talk about dogs. Dogs are cool. I think it might just be me. Okay, let's see. I'll dispel some dog myths right now. Dogs with underbites are not better at biting. That's a myth. Dog teeth are designed to work as an interlocking scissor, and if they don't work that way, they're less effective, no matter what bulldog breeders tell you. Daria's not making any sounds either.
Poopy farts.
Oh, now here we go.
I was checking my tobacco because I'm growing some tobacco seeds and I completely forgot and I haven't watered them in a while, so I had to check. And
How are they coming along?
they're growing. They're really tiny. Like tobacco seeds are absolutely tiny.
A mustard seed
So
kind of situation. Yeah, I've got some coming also. I'm going to be growing some as well because I've got some agrobacterium and some
mm.
plasmid coming.
I just like want to grow stuff hydroponically because we're kind of having problems with our you know farming and everything, and the world kind of depends on Ukrainian farming. So just
You know,
experimenting with hydroponics.
that's interesting. I think I was at Disney one time years ago, and they had aerial aeroponics or something they were doing, where they just have like plants with the roots just dangling, and they just like spray, you know,
Mhm.
fortified water on them basically, and then they kind of go through a thing and get sprayed again here in a minute. Like I don't know if there's any advantage to that. It seems like it's a lot more work than just sticking them in dirt, but it was interesting that plants can survive that way.
Yeah, I have like a little DIY hydroponics station here.
Oh, that's cool. Yeah. Do you have to worry about contamination with that? I mean, I imagine plant roots don't really care if
In
it's.
hydroponics, not really. It's like a just a plant. You have the water circulating all the time because there's
Yeah,
different
I
levels.
guess the
But
media
like
is.
I did contaminate when I just have like a glass petri dish here, and I had the seeds. So like when I was trying to grow salad, like
Yeah,
I open it and there's just mold everywhere, and I'm like, ugh.
yeah, yeah. Mold is such a nightmare sometimes.
Yeah, it'd be pretty exciting to start working with plants
Yeah,
again. We were
yeah.
working with cannabis plants a long time ago at the Odin. Um, but since then, we kind of Texas cannabis isn't legal yet, so
Yeah, I suspect that'll change in a year or two, though. I really do.
Yeah, come on, Texas, come on.
But yeah, I think I think it'll be a lot of fun working with genetically modified plants. I'm excited. It's like a
yeah,
whole new world to me because like I've specialized in animals for so long, and then like bacteria because you have to, and so like you know fungus and plants is like a whole different universe for me.
oh yeah,
But yeah, oh, something went wrong with the chat. Looks like.
Yeah, I also saw it.
no, nothing went wrong. I just blocked somebody who said something really,
Yeah, I thought so. Yeah, yeah, good.
said something pretty,
Pretty sexist. Yeah.
you know.
Uh, don't know. Yeah, developing a good bullshit detector is critical for pretty much anything.
Yeah.
Generally, just don't like shitting on other people specifically.
Yeah.
So
But I think the biggest thing is just like learn as much like basic biology as you can. Because usually when people just make up shit, it violates a lot of like things that make like common sense
violates
argument.
thermodynamics.
Yeah, exactly. Right. Just like simple
If
stuff.
anybody says, I mean, there's a couple basic things. If anybody says this thing cures everything or cures like way too many things,
Yeah.
it's definitely not true, right?
Yeah.
If somebody says something that violates the laws of physics or thermodynamics or something like that, not true, right?
Yeah.
And like generally, the chances of some rando um, proving a bunch of scientists wrong is almost never going to happen. So don't believe them or have stringent requirements on what you accept as something that would prove, them correct.
Yeah, don't just be like, ah, you made one YouTube video, let me just believe you. Yeah, definitely like, have standards of evidence.
Yeah.
Because like it's supremely easy to lie. It's much harder to prove things.
Yeah, it's easy for somebody just to say like, "Look, I am now not lactose intolerant. I can eat pizza." Um,
Ooh, shade.
to say stuff like that. It's a lot harder to show through gene expression and through, stuff like that. So usually when somebody's saying something and they're not backing it up with any sort of data, like real data, then
Yeah.
you probably know that even if it is true, like you have no evidence.
Yeah.
Yeah.
And also, and that includes us. Like that's why we always go out of our way to like, provide people with evidence for all the things that we do and every step of the way, because
Yeah. We
that's a good
always
example for how you should be.
Yeah, we always show all the work we're doing, We show
And
pictures of all the stuff. We show exactly talk about exactly what we're adding, what we're doing, and everything, And so we try to be as open and transparent as possible. And when people aren't open and transparent, when people are like, well, it's proprietary or like I'm trying to patent it or something, no. I mean, like, come on.
yeah, and that's the thing, because it's like you know you have to just, and also like it's important to like be honest when you fail, right? It's like, we fail a lot. We try a lot more things than we succeed at, because biology's hard. And so you just, but it's important to be like I think a lot of people are like, oh no, if I fail, everybody will lose respect for me. But I think it's the other way around. If you lie about your failures and pretend you succeeded, I lose all respect for you. You gotta like, be honest, because like failure is a part of science.
Yeah. The thing is like generally everybody in the world has a pretty good bullshit detector, and so like even if you do lie and convince some people, like the majority of people aren't, you're not going to have anything for them. So it's just like,
Yeah.
it's one of the ways that like we've gotten so popular. We've had people write about us in books and everything like that, as we're open and transparent and we don't bullshit, And it's like
Yeah.
anybody else can come up against that and ask us like, well, what is your protocol or where's your data, and we could immediately point them to it and be like, here's our data, here's the data we took. You want to look at our COVID vaccine? Maybe I shouldn't mention that in streams,
Yeah.
but just look at the data. Go to this the-cdc.com and you can find links to our data and all the experiments we did and protocols. Like
Yeah.
that's how people have it. Even normal academic scientists don't have stuff like that. And so it's
Yeah,
like,
we have videos, which some of them may not exist anymore because of certain platforms. But we had videos of like us doing the assays and stuff, so you can like watch us do the assays, which nobody has that. But I feel like if we do that enough, maybe that'll become like the standard for what's expected of biohackers, is to like back your shit up, and then people will uh, you know will feel like, oh, if I don't back my shit up, people will not believe me, and then there'll be a good set of data and honesty and transparency coming out of the biohacking community in general over time.
yeah, I just think you don't get anything by shitting on specific people. Like you don't win. Like there's nothing to gain from it is the thing. And so like doing it actually is like there's no reason. So it's yeah, sure we talk shit about the government and about academics in more of a general sense, but there's good people in the government, there's good academics. Um, and so it's just like, you know, uh, we try to focus on the things that we can do,
Yeah.
and not what everybody else is doing wrong.
Yeah. Yeah, and I think that's a lot more powerful to try and be a good example than it is to just go get in fights all the time.
Yeah, I don't know. Making YouTube videos talking trash about people just seems like so childish.
Yeah, for sure.
Come on, it's fun sometimes to talk shit about people.
What are you, 13? Sure, like in private when I'm drunk, but like I don't. Who am I? What am I trying to do? Am I trying to make somebody have a bad life? Like that seems like a shitty human being who's like trying to ruin somebody's source of income or something like that.
No,
Like I
yeah.
don't, even if you're a grifter, like fuck, like you still have kids and you need to feed them. I'm sure. Like
Yeah.
I'm not trying to ruin anybody's life. That's like what fucking fascists do.
Yeah, not to mention, people are generally, if you're full of shit long enough, people will figure it out.
Yeah, karma comes and gets you.
Yeah.
Make a drunk biohacking stream. We would.
Just come to Biohack the Planet. I'm sure there will be.
Yeah,
some
come to
content.
Biohack the Planet, which is come to Biohack the Planet, which is the beginning of September. Just search Biohack the Planet 2022 and online, and you'll get a link. It's going to be a lot of fun. We're going to be drinking. We're going to be doing science. It's going to be a really good time.
Yeah, you can even take a picture with a living Ukrainian refugee,
You always,
like in a zoo.
Are there people like visiting, just going around being like, "Hey, come take a picture with me so I can show people how good it
No,
is"?
but they're like, I mean, there's a lot of foreigners right now, but usually most of them are reporters or volunteers or instructors or stuff like that. But like, I've seen people on the internet being like, yes, I just like people would be like, I don't know, people like Lex Fridman. You probably heard about him. He's like a big podcaster, and he
yeah,
like at first he like made a podcast with like a literally a dude who like feeds Russian propaganda straight up, but being like, I'm trying to be objective and like not to
oh
take
god,
sides, and it just literally spit out Russian propaganda. And then he
Wow.
went to Ukraine, and like then people was like, oh, I learned so much about how horrible war is. I learned so much. I am so humble right now. And you're like, shut the fuck up, because like we're not animals to like take pictures with us and being like, I don't know, and like taking selfies in front of ruined buildings where people died. Like this is not a game. This is not a zoo.
Seems
Like
really
there's a
yeah,
tragedy. Like it's important to spread information about what's going on, but like don't make it an entertainment.
yeah,
yeah.
I agree. Seems gross.
Yay! Someone was. Thanks, Robin.
Hmm.
Yeah.
I want to take pictures with you, Daria. Come on.
$5 for one picture.
What? Fine.
What? It's exotic. Come on, Ukrainian refugee in Texas.
I'm sure I could find other ones. I think I
Okay,
know of
I'll trade you a picture with an armadillo.
others. Yeah,
Armadillos.
I'll trade you.
Oh my god,
I know how much you love armadillos.
I love armadillos. Okay, I'm coming to buy Hug Planet. At first, I was like, okay, there's a 50-50 chance I'm coming, but now it's a 90% chance because of the armadillos.
Okay. I'll trap an armadillo and have it here waiting for you. Just so you can pet an armadillo.
But they're so fast.
Armadillo petting zoo.
I'm
We'll
not
rent
Texas.
those in the zoo.
I can definitely catch an armadillo.
There's got to be a
No.
place in Austin that lets you rent armadillos. Like,
I
100%.
honestly would be surprised if there wasn't. It is Texas.
Renting an armadillo? That's like the most Texan shit ever.
I'm sure there's a petting zoo somewhere that has a petable armadillo.
Yeah, there should be like zoos where you can pet.
I saw one
Oh
where
my
you can
God.
like swim with otters and pet the otters and stuff in Louisiana. I was like, that's on the list. We're petting some otters
So
one day.
cute. Because I remember I was chasing that armadillo like in front of the Odin building in the parking lot, but it was really fast. Like I couldn't catch up with it.
Your legs are this long.
I know the feet are like little feeties. Oh. Oh
But yeah,
no, not armadillo stem cells. Come on.
Biohack
Armadillos are
Planet,
secret.
it's going to be a really good time. And it's really small this year. We're making it really small so it's intimate so we all get to hang out and just like have a good time.
Are there any
Next
tickets
year,
left?
there's like 6 tickets left.
Hmm.
Something like that. Yeah. So 6 tickets. Make sure you get your tickets soon. They're going to be gone. But yeah, it's going to be a really great time. We're going to have workshops on stem cells, microinjection.
Maybe
And,
maybe I'll get this.
we're going to do plant stuff, mushroom stuff. It's going to be really cool. Maybe even mushroom genetic engineering. That would be really fun. Fungi,
Can we just
something
do
Bear and I have been
mushrooms?
working on. Do actual mushrooms.
Yes.
No, but they do have that weird THC that's legal, Delta 8. So we're probably going to get some Delta 8 THC so people can, get high if they want. Yeah. It'll be all around just like a biohacker party, learning science, doing science, having a good time. Yeah.
Yeah, it ought to be a lot of fun. I've never been to a biohack planet that wasn't fun, and I was super disappointed that 2020 and 2021 didn't
Yeah.
go.
Yeah.
Damn pandemic.
Yeah. So, yeah, I know. What was the last one? 2019?
Yeah.
2019.
We tried to do one in 2020, but then they wouldn't refund us, but eventually did. And then 2021, I was just like, no.
Just move.
Some people tried to schedule conferences in 2021, but yeah, no. So yeah, it's finally time. People are being allowed to do stuff again, and yeah.
Yeah. And even that, we're starting kind of small, and then, maybe 2023 we'll do something a little bigger or something.
Yeah. Yeah, I think so. Next year we'll probably have more of a conference. Yeah.
And I'm looking forward to
Anyway,
this
make
one.
sure you make it. Yeah, like I said, there's still a few tickets left, but I'm sure they'll go soon.
Yeah.
If you need like a discount or something, just contact us and we can try and help you out to make sure you just get there. It'll be really fun. But otherwise, thanks everybody for coming to the chat today, live stream. It was fun. Next week we'll be back, same time. Keep updating you on our stem cell projects and other
The
projects.
period project. What is it? The blood science?
Oh, no.
No, you won't have that. That's 2 more weeks.
Yeah, yeah, true. What is it? Uterus something. It was really funny. Yeah, Robin, you missed
it was
all the
period.
fun.
But yeah, thanks everybody for coming. Support us on Patreon. We all got Patreons. Go just search our name in Patreon and you'll find something. And otherwise, we will see you next week. Same place. CDC rules for life.
Bye.
We need like a some sort of like hand sign. CDC.
What are you doing?
We'll have to work on it. It won't be cool if it won't be cool. Nope, it's not cool.